Welcome to the OBU Hackathon. My name is Riley Davis and I'm a biomedical sciences major.
My name is Rebecca Yank and I am a biology major with a minor in computer sciences.
I'm Michelle Phillips and I'm a biology education major.
We're so excited that you're here with us today, and we are also excited to guide you through what it means to annotate a phage using bioinformatics.
In this video, we're going to tell you what a phage is, how it matters in the real world, and we're going to give you a brief overview of what annotation looks like.
Okay.
Hey guys, the goal of the hackathon is for you to learn how to annotate a genome. We're going to start with a background information on what exactly is a phage. So by definition, a phage is a virus that destroys bacteria by lysis. Viruses are very common, and the most common that y'all all know of is COVID-19. So we're all familiar with viruses. These phages destroy bacteria by lysis, which is the disintegration of a cell by the rupture of the cell wall or the cell membrane. There are thousands of these phages in the world, and we're still discovering more today.
So why does bioinformatics matter? It matters because research is important, and we as scientists are constantly expanding our knowledge of what we know and seeking answers to problems that we have. Some of the problems that we are currently having is in the medical field. We are looking for ways to treat different bacteria that have become resistant to antibiotics, and a way to do that is using phages. So because phages are host-specific, doctors are able to create phage cocktails to treat specific types of bacteria that have become resistant to the antibiotics we already have. The way that they find these phages is using phage libraries. The way that these libraries are assembled is through researchers and students like you who annotate genomes of phages that they find.
So what exactly is annotating? At OBU, students find phages in the environment. I found one in my backyard, and work with them in the lab by allowing them to infect bacteria on plates. Then we send the phages off for sequencing, which gives us all of the nucleotides in the phage's DNA, which is also known as the phage genome. When we first get this information, it's just a bunch of letters strung together with no meaning. Our job is to annotate the genome by finding out where genes are and what their functions are. We use all kinds of different software to help us accomplish this goal. We have a computer program go through the genome and predict where it thinks genes are. Then we have to check the program and see if its predictions are correct. Ultimately, there are two questions to keep in mind when annotating. The first one is, where is the start location of this gene? And the second one is, what is the function of the protein produced by this gene? In your search to the answers to these questions, treat it like you're a lawyer making a case for your gene. We have to gather all the possible evidence to prove that our predictions for annotation are correct. We are excited to work with you on your annotation journey, and you will be so amazed at what you can accomplish when you're done.